Hi, everybody. Welcome to the lecture that covers Chapter 4, How Cells Obtain Energy, as well as Chapter 5, Photosynthesis, which we are covering together this week because those two chapters go hand in hand with each other. We're going to start this lecture off by asking you to think about what you ate for breakfast today, if you ate breakfast. Maybe you had eggs and bacon, maybe you had yogurt and fruit, maybe you had a bowl of cereal or something convenient like a protein shake. These foods are all quite different from each other, but what they have in common is that they provide you with some combination of the three nutritional macromolecules, which are carbohydrates, lipids or fats, and proteins. And remember, the fourth class of macromolecules is nucleic acids, but as we talked about in a previous lecture, the nutritional value of nucleic acids like DNA is negligible, and so they don't really contribute to nutrition. So, carbs, fats, and proteins are the things that we're talking about that fuel you and provide you with energy. And as a living organism, you need energy. This is one of the characteristics that unifies living organisms and that we talked about in our first chapter. Energy processing is one of the characteristics that all living organisms possess. And the reason why living organisms need to process energy is in order to drive their metabolism. Metabolism is probably a word that you've heard before, but the scientific definition of metabolism is that it is the sum of all chemical processes occurring inside of living organisms. So it's not just the process of digesting your food, it's everything that's going on inside of your body. And organisms need energy for just about everything that they do in their metabolism. This is regardless of whether they are a plant or an animal or a microscopic organism or any other type of living thing. They need energy in order to break down large molecules into smaller ones. They also need energy to build up large molecules from smaller ones. They need energy in order to transport materials in and out of cells and around the body system. They need energy to destroy pathogens or invaders that could cause infections and cause destruction, and many other things. So, together, all of these processes are called metabolism and they need energy. So, this lecture is all about how organisms acquire that energy. And when we're speaking about energy, we need to be careful to define what energy really means because there's a lot of sort of colloquial common language understandings of what energy is that may not be accurate to what we're talking about in biology. The definition of energy that we are using here in this class and for our purposes is actually a definition of energy that comes to us from a field called thermodynamics. And thermodynamics is the area of chemistry and science that studies energy and energy changes. According to thermodynamics, energy is defined as the capacity to do work. Also, according to thermodynamics, energy, or the capacity to do work, can exist in one of two different states. Kinetic energy is the form of energy where that energy is actively being used in motion. Potential energy is the alternative form of energy, where that energy is being stored, but it is not actively being used in motion yet. If we look at the picture on the screen here, we see water that is flowing over the edge of a dam. And so the water that is falling, the water that is actively in free fall over the edge, is exhibiting kinetic energy, the energy of motion, while the water that is located at the top of the dam is exhibiting potential energy because it has the potential to fall over the edge of the dam and therefore enter into a kinetic energy state. But right now, that potential is just that. It's only a potential. And so we consider that to be stored energy or the stored capacity to do the work of falling. Obviously, then from this example, energy can be converted between these two states because the potential energy of water that is located at the top of the dam turns into kinetic energy the moment that it flows over the edge. So the overall goal of metabolism has to do with converting potential energy found in nutritional molecules into kinetic energy that can be used by organisms to do all the things that they need to do. Found within the fats, the carbohydrates, the proteins that you eat is an enormous amount of potential stored energy. And metabolism seeks to release that energy so that you can then use it for your purposes. In molecules like carbohydrates, fats, and proteins, the potential energy is stored inside of the chemical bonds between atoms in the molecule. It is the bonds that house all of the energy. If we look at the picture on the screen right here, just to refresh our memories, we see an atom of carbon that is in covalent bonds with 4 atoms of hydrogen. And the covalent bond means that it is actually sharing its electrons with the hydrogen atoms here. So those shared electrons represent the bond. Therefore, as we already know, chemical bonds themselves are composed of electrons. So if bonds are made of electrons and it is the bonds that hold the potential energy, this should make some sense to us. Because one thing that you may also already know about electrons is that electrons carry electricity or energy in other contexts as well. So the electrons that run through our wires carry energy to our light bulbs, our air conditioners, our microwaves. All of these things are powered by the energy of electrons. And so it should make sense to us that it is the bonds in the chemicals, i.e., the electrons, that are storing the energy in a molecule. However, not every bond within a molecule is equal in terms of its energy. So for example, we're looking right here at a molecule of the sugar glucose. We can see that it's made up of a combination of carbon, oxygen, and hydrogen atoms put together in this hexagon arrangement. One of the 3 different types of bonds that you see in this molecule is a carbon bonded to a hydrogen. That particular type of bond stores 98.9 calories per mole. And a mole is a measurement of quantity used in chemistry. You don't have to know for our purposes what it means, but we kind of think of it like a dozen. It's a measurement of quantity. So 98.9 calories per mole in a carbon to hydrogen bond. We also see in this molecule that we have carbon to oxygen bonds. And those type of bonds hold 85.6 calories per mole. And we also see that we have carbon bonded to additional carbons, and those types of bonds hold 82.9 calories per mole. So a different amount of energy, a different amount of calories is stored in different types of chemical bonds. Therefore, it makes sense that, because every molecule is structured differently, different molecules contain different amounts of energy that can be derived by living organisms. So overall, if you take all of the chemical bonds together in a molecule of glucose, together they provide 686 calories per mole, while down here at the bottom of the screen, we see a molecule of palmitic acid, which is the primary fatty acid found in the lipids that make up corn oil. Palmitic acid, on the other hand, provides 2,430 calories per mole. So it's a lot more energy dense than the glucose molecule, and this explains why some types of macromolecules are more energy dense than others. Or, in other words, why some of them provide you with more calories per gram than others do. So, how do our bodies and our cells and the cells of all living organisms actually access and harvest the chemical energy found in these molecules? Well, in order to get at the chemical energy located in those stable bonds, you actually have to break those bonds apart. Breaking down the chemical bonds in these molecules releases the energy that is contained in the electrons that make up those bonds. And at that point, once the bond is broken, then the energy can be transferred into a more easily accessible form. That easily accessible form comes in the form of another molecule called adenosine triphosphate, or ATP. ATP is a very important molecule for all living cells because it is considered the energy currency of the cell. ATP is special because although it is a relatively small molecule, it stores a large amount of energy, and most importantly, it stores that energy in a way that is easily accessible to the cell. And I'll give you an analogy for this. We know that sugars, carbohydrates, and fats store a large amount of chemical potential energy. But that energy is not easily accessible to the cell for all of its purposes. Adenosine triphosphate stores large amounts of chemical energy in a way that can be deployed at a moment's notice for any purpose. So we can think about sugars, carbs, fats, proteins as being like gold bars or precious jewels or a cryptocurrency like Bitcoin. All of these things hold value, but you can't walk into a store and hand them a gold bar and expect to pay for your groceries. It's just not useful. Adenosine triphosphate we can think of as being like cold hard cash. It can be used any place, anywhere, for any transaction to transfer energy for any purpose. And that's why ATP is a powerful molecule to the cell. So let's take a look at this molecule ATP and figure out how it is able to store so much energy despite the fact that it is small. If we look closely, we can see that the structure of ATP includes 3 phosphate groups along the left-hand side of the molecule as shown here. Those phosphate groups are all bonded to each other, and they all have a negative charge associated with the oxygen on the phosphate group. You can see that in the little negative sign that is located right here, here, and here. One thing that we know about negative charges is that while they are attracted to positive charges, they are repulsed and repelled by other negative charges. And so you can imagine that the effort and the energy involved in keeping 3 negatively charged molecules directly adjacent to each other, that's a lot of energy, right? If you've ever tried to hold the North Pole of a magnet up against the North Pole of another magnet, it's really hard to keep them together in close proximity. They physically repel each other. And so the bonds that are located in between these 3 phosphate groups are actually very high energy bonds because they have to be in order to keep these negatively charged groups together with each other. So that's how ATP is able to store a lot of energy. And again, the way that that energy can be released for other purposes inside of the cell is by breaking the bond between the phosphate groups, which allows the phosphate group to come flying off, and all of the energy that was previously being used to keep it in place is now available for some other purpose inside of the cell. When that happens, when the bond between the second and the third phosphate group is broken, then adenosine triphosphate is no longer adenosine triphosphate. Instead, it becomes what we call adenosine diphosphate. It was called triphosphate in the first place because tri, meaning 3, was indicative of the fact that it had 3 phosphate groups attached to it. But once you break off that last one in order to access its energy, it turns into adenosine diphosphate. And the di indicates that now it only has 2 phosphate groups here. That energy can be used for some other purpose in the cell, but the beauty of this molecule, ATP, is that once it becomes adenosine diphosphate, ADP, it can be recharged back up to adenosine triphosphate by harvesting energy from some other nutritional macromolecule. So we can think about ATP as being like the fully charged version of the molecule, and we can think about ADP as being like the version that is almost out of charge. And we can bring it right back up to ATP if we just attach another phosphate group here. So that's the goal of metabolism. The goal is to take chemical potential energy found in molecules like carbs, lipids, and proteins, which are very valuable and rich in terms of energy, but that energy is not easily accessible. And we take that more long-term potential energy and convert it into a short-term form of potential energy, which is the molecules of ATP, which again are also rich in energy, but that energy is useful. And then that short-term potential energy can then be deployed and used to convert to kinetic energy for organisms to carry out all the processes that they need to do. Now, in this lecture, we don't have the time to talk about the metabolic breakdown of carbs, lipids, and proteins altogether. So we're going to limit the scope of our investigation here, and instead we're just going to talk about the metabolic breakdown of sugar molecules by your body. In this first checkpoint, what I want you to do is tell me what is the biological macromolecule that sugars are the monomers of. This is a review from a previous chapter that we covered, but tell me, sugars are the monomer of which of the 4 biological macromolecules? Now, I haven't mentioned it yet, but there's a name for this process by which chemical potential energy is harvested from sugars and converted into ATP. And that name is cellular respiration. Cellular respiration is the name of the whole metabolic process that we are talking about here. Cellular respiration is a complex process that can be divided into 3 separate steps. The first step is a chemical reaction called glycolysis. The next one is a series of chemical reactions called the citric acid cycle. And then finally, the third step, which yields the most ATP, is called oxidative phosphorylation. Let's take a step back for a second and talk about where this stuff is happening inside of your cells. Glycolysis is the only one of the 3 steps of this process that occurs in the cytoplasm of the cell, which remember that the cytoplasm is the jelly-like fluid substance that makes up the inside of the cell. If there's dead space in the cell, it is filled with cytoplasm. The citric acid cycle and oxidative phosphorylation both occur inside of the mitochondria, which we previously learned is an organelle or a cell structure that is dedicated to generating energy in the form of ATP. So now we make that connection. If we take a closer look at the anatomy of a mitochondrion, then we can see that unlike in the electron micrograph image right here, this cartoon image points out to us that there are 2 separate membranes clearly delineated. There's an outer membrane, which forms the sort of bean shape, and then an inner membrane that is consistent of a bunch of different folds here. The inner part of the mitochondrion is called the matrix here, and the folds in the membrane, the inner membrane, are called cristae. So we're going to walk through the 3 major steps of cellular respiration: glycolysis, the citric acid cycle, and oxidative phosphorylation. And keep in mind as we're doing that the first one will occur in the cytoplasm, but after that the process migrates into the mitochondrion where the last 2 steps take place. So we're going to dive into this process and walk through the 3 steps of cellular respiration, starting with glycolysis. Glycolysis starts with a glucose molecule and carries out chemical reactions that essentially break it in half. The glucose molecule that you see on the left-hand side of the screen turns into 2 molecules of what is called pyruvic acid. And the pyruvic acid looks like this. As you can see, or as you can imagine, if you took 2 of these and fit them together with each other, you would get something that looks like the glucose hexagon, roughly. In the process of doing this, glycolysis releases enough energy from the bonds in the glucose molecule to recharge two molecules of adenosine diphosphate, which remember is the uncharged version of the energy currency, back up to the fully charged version of adenosine triphosphate. So it can do that 2 times. In addition, something else happens, and that is that the cell takes 2 molecules of a compound called NAD+ and converts them into 2 molecules of NADH. Now, NAD, I don't want you to know what it stands for. It stands for nicotinamide adenine diphosphate, but that's not something I want you to know. What I want you to know is that NAD and NADH are the uncharged and charged versions of a molecule that is even more temporary of an energy carrier than ATP. And at the end of this process, what we're going to see is that the NADH molecules are eventually going to have their energy taken and converted into ATP molecules. So we're going to see some collection of NADH molecules along the way as well, but know that eventually they are going to be converted into ATP energy. So at this point, after we've completed the first step of cellular respiration, glycolysis, the cell has harvested 2 ATPs from this glucose molecule by breaking it in half, as well as 2 of those even more temporary NADHs, which will come back into the picture later. We now see that we have 2 pyruvic acid molecules down here, and there is still more energy located in the bonds of the pyruvic acids, so they can be broken down even further. They are broken down even further in the next step of the process, which is called the citric acid cycle. So the citric acid cycle starts by taking the 2 pyruvic acid molecules, and each of them is converted into a molecule called acetyl-CoA, which looks like this. In the process of converting them into these molecules of acetyl-CoA, some of the pyruvic acid gets broken off and lost. In fact, it's lost in the form of a molecule that we're more familiar with called CO2, carbon dioxide. And what do you suppose happens to that CO2 molecule? Well, it gets exhaled. It gets released from the organism as a waste product. And that is the reason why organisms like ourselves exhale and release CO2. In this process of converting pyruvic acid to acetyl-CoA, no ATP is created. However, one of those even more temporary energy carriers, the molecules of NAD, gets charged up and turns into an NADH. After this happens, the acetyl-CoA enters what is a series of 8 chemical reactions that take place in a cyclical manner that are called altogether the citric acid cycle, named for the fact that one of the intermediate chemicals that pyruvic acid ends up getting converted into is citric acid. In fact, that's the first step of the cycle. That's why it's called the citric acid cycle. I don't care if you know what these chemicals are, but what I do want you to know is that over the course of this process, the acetyl-CoA is completely broken down and torn apart into molecules of CO2, which again are released as a waste product. And from this process of completely breaking down the acetyl-CoA, more energy is harvested. Specifically, for every 1 acetyl-CoA molecule that goes through this cyclical chemical breakdown, 3 molecules of NAD are charged up into their form of NADH. Another temporary energy carrier called FAD, or flavin adenine dinucleotide, again, don't want you to know that, is charged up into its fully energized form of FADH2. And then finally, 1 molecule of ADP is charged up into a molecule of ATP. Now, when you consider the fact that in glycolysis, we saw glucose get broken down into two pyruvic acids when it was split in half, we know that for every glucose molecule that starts this process off, everything that you see on the screen here must happen twice because there are two pyruvic acids that come out of glycolysis. Therefore, if we consider the total amount of energy that is harvested from the citric acid cycle, at this point, we have a running total of 4 ATP molecules. Remember, 2 of those came from glycolysis, and the other 2 would come from the citric acid cycle running twice, once for each pyruvic acid that emerges from glycolysis. We also have a running total of 10 NADH molecules, which probably sounds like a lot, but remember, 2 of those were made in glycolysis. Another 2 come from the initial step of converting pyruvic acid into acetyl-CoA, which happens twice because 2 pyruvic acids come from glycolysis. And then finally, another 6 come from the citric acid cycle running twice to break down acetyl-CoA, and each of those times 3 is produced. So therefore, 6 plus 2 plus 2 from glycolysis gives us a running total of 10. And then finally, we have 2 of these molecules of FAD, FADH2 that were created, 1 per citric acid cycle, 2 total because of the 2 pyruvic acids. So at the end of the second step of cellular respiration, we've only made 4 ATP, but we've made a lot of these other temporary energy carriers. We've made a lot of NADH and FADH2. What happens, as promised, in the third and final step of cellular respiration is that temporary energy runs through a series of chemical reactions that converts it all into ATP. And as you can see, in this final step, what you get is a landslide of ATP molecules compared to what was harvested in the first 2 steps. 34 ATP molecules, bringing the grand total of ATP molecules made during this process to 38. Oxidative phosphorylation, in the process of making this conversion, consumes 6 molecules of oxygen. Where does that oxygen come from? It's the oxygen that you breathe in. This is why you need oxygen. This is the very reason why it is necessary for organisms like yourself to inhale oxygen, because it is used in oxidative phosphorylation to convert NADH and FADH2 into ATP molecules. That oxygen ends up getting converted into water in this process, and that water goes on to just become part of the normal water that is found in all cells in your body. So we just finished talking about the grand total number of ATP molecules that would be created as a result of a glucose molecule being broken down over the course of these 3 steps of cellular respiration. In this checkpoint, what I want you to tell me is how many ATP molecules would be produced if 2 molecules of glucose sugar were completely broken down in cellular respiration. Not 1, but 2 glucose sugar molecules. Now, as we wrap up this chapter, we're going to take a step back and sort of look at the big picture of what's happening in cellular respiration as an organism breaks down this sugar to extract its energy. What you see on the screen here is a chemical equation, which shows on the left-hand side of the arrow all of the things that are considered an input into this process, and on the right-hand side of the arrow, all of the things that are considered an output. We know that glucose is the thing being broken down, but another ingredient is required in this process, and that's the oxygen that we saw active during the third and final step of oxidative phosphorylation. Those 2 ingredients together yield, out of the reaction, a large amount of waste carbon dioxide that is released when you exhale, some water that becomes part of your normal water that makes up your body, and then a large amount of ATP energy, which is derived from the chemical bonds in the glucose molecule. So tell me really quickly here, recall which of the three stages of cellular respiration is the one in which the carbon dioxide is released? Which of the three stages generates that waste carbon dioxide? So at this point, we're going to transition over from chapter 4 into chapter 5, which is about photosynthesis, a companion process to the one that we just talked about, cellular respiration. Our discussion is going to remain along the same lines as we've been talking about so far, which is why we clustered the two of these chapters together. But at this point, we're going to take a further step back and ask the question, where do organisms get the molecules from which they harvest energy? We just finished discussing and going over what is involved in breaking down a molecule of sugar in order to extract chemical energy in the form of ATP. But where does that sugar come from in the first place? We have two organisms here on the slide. We have an animal and we have a plant. And as you probably know, plants are not like us in terms of how they get their chemical energy in the first place. Plants are able to take the energy of sunlight and convert it into those nutritional macromolecules, the carbohydrates, proteins, and lipids. Animals don't do this. Instead, they eat the nutritional macromolecules that are provided by plants. There are two different classifications of organisms based upon how they harvest their energy. Plants are considered autotrophs, or in other words, producers. If you think about what the prefix auto means, it means self, and therefore autotroph means that the plant is capable of producing its own sugars, its own macromolecules. And it does this by harvesting the kinetic energy of sunlight and converting it into chemical potential energy for themselves. Heterotrophs, or in other words, consumers, are like us. They are animals. They cannot perform the process that plants do by capturing sunlight energy. So instead, heterotrophs have to consume the chemical energy that is produced by autotrophs. Now, if we go back to that question of what it is you ate for breakfast today, and we rethink about all the options that you had on the table there. We had bacon and eggs, we had yogurt, we had berries, cereal, protein shake. All of these things have those nutritional macromolecules in them. But if we think about where they came from, we think about their true origins. Like, let's pick one out. Let's talk about those berries. Berries come from a plant, right? And so that plant grew based upon its consumption of the energy of sunlight. So when you're eating the berries, you're actually eating energy that used to be energy of the sun. What about the bacon and eggs? Well, the eggs were laid by a chicken, and that chicken, although it is an animal, it had to eat grains and plants in order to survive, which those grains and plants that serves as the fuel source for the chicken obtained their energy from sunlight. So it's a few steps more removed, but the eggs too contain the energy of sunlight. I think you get the idea of what I'm getting at, which is that energy flows in one direction from the producers to the consumers in the biological world. And if we trace all these types of chemical energy back to their original source, the original source of energy in the biological world is the sun. The sun's energy is captured by the autotrophs, or in other words, the producers, and then through the autotrophs being consumed by the heterotrophs, is able to pass to these other levels of consumers. All of this is to say that even if you're not a plant, the sun is extremely important to your livelihood because it is through organisms like plants, the autotrophs, the producers, that energy is able to be captured and turned into a form that can be consumed by the heterotrophs. So Right here, what you're looking at is a graphical representation of what we just learned about in Chapter 4. We saw the process known as cellular respiration, which is the process by which sugar molecules are converted into a ATP molecule, which can then be used for all sorts of kinetic processes. How those sugar molecules arise in the first place is they are produced by autotrophs, such as plants, in a process called photosynthesis. Photosynthesis literally means to produce things using the energy of the sun. That is what the word photosynthesis means. And so, whereas plants perform photosynthesis to create sugars, and then they break those sugars down through cellular respiration for their own uses, organisms like animals, like ourselves, the heterotrophs, get on the bus a stop later, as it were. Instead of making the sugars themselves, they effectively steal them from the autotrophs and break them down through cellular respiration. So bottom line is that while both plants and animals, while both autotrophs and heterotrophs perform cellular respiration to break down sugars, only the plants make those sugars themselves. The heterotrophs, the animals, do not do that. Now, I've been referencing plants, but plants are not actually the only type of organism capable of performing photosynthesis. There are also certain types of bacteria, called cyanobacteria, that are capable of photosynthesis, as well as aquatic algae. And here you see a type of seaweed pictured, and seaweed is actually considered to be an algae. And as we move forward and talk about how photosynthesis works, we are going to focus on how photosynthesis works in aquatic algae and land plants. And the reason why we're leaving photosynthetic bacteria out is because photosynthesis looks a little bit different in photosynthetic bacteria. Because, as you know, bacteria are a prokaryotic cell, which are much smaller and more simple, and they don't have internal membrane-bound organelles. Aquatic algae and land plants do have a specialized organelle in which photosynthesis takes place, and that organelle is called the chloroplast. Chloroplast we learned about previously when we were talking about cell structures. Here's an image of what chloroplasts look like under the microscope. You can see all the individual plant cells that are lined up next to each other, kind of like bricks in a wall. And inside them, there are little green orbs, and those green orbs are the chloroplasts. If we could get an even closer look at the chloroplast, then we would see that the anatomy is sort of complex. It also, like the mitochondria, has two layers of membranes. You may remember that the mitochondria had an outer membrane and then sort of a squiggly inner membrane. The chloroplast has two smooth membranes, one outer and one inner. And then within the inner membrane, there are these stacks of structures called thylakoids. And these stacks kind of look like green stacks of pancakes, and they're surrounded by material called stroma, which is the liquidy jelly-like substance that fills up the inside of the chloroplast. We divide photosynthesis into two separate stages, and as we talk about those stages, we're going to talk about the part of the process that occurs either in the stroma or in the thylakoids. So just be aware of the anatomy of the chloroplast for those purposes. Before we start talking about the details of photosynthesis, we're going to look at the big picture of the process by once again looking at one of these chemical equations that represents overall what are the inputs or the ingredients that are going into the process and what do those get turned into. A lot of this is very intuitive for you already. You probably already have a sense of what plants need in order to perform photosynthesis. So if we look on the left-hand side of the arrow, we can see that energy is represented here. That, of course, would be the energy of sunlight. We also see that plants consume carbon dioxide as one of the ingredients in this process. And they also consume water. And this is easy to remember. You know these things already, most likely. On the other side of the arrow, what does this all get turned into? Well, it gets turned into oxygen, which is given off through the process of photosynthesis, as well as molecules of glucose sugar. Now, if you remember what we just talked about with respect to cellular respiration, this equation might look familiar to you. And indeed, it is, because the overall equation representing the ingredients and the products of cellular respiration is exactly the reverse of the recipe for photosynthesis. Anything that is a product of cellular respiration is an ingredient of photosynthesis, and vice versa. So if you understand and know the overall equation for one of the processes, you understand and know the overall equation for the other one. So now that we've seen what goes in and out of photosynthesis, let's take a look at the inside of a chloroplast. Let's talk details about the steps involved in the process. We saw that cellular respiration was divided up into 3 separate phases. Photosynthesis is divided up into only 2. One of them is called the light reactions, and that one happens first. And the second one is called the Calvin cycle, and that one happens second. So we're going to talk about these stages one by one and what goes on in them, and how the ingredients are taken in and then crafted into sugar and oxygen molecules. We'll start with the light reactions here. So here we've zoomed in this figure on just the light reactions, and you can probably see that they are localized in one of those green stacks of pancakes called the thylakoids. Specifically, they happen in the membrane that surrounds the thylakoids. Like their name suggests, the light reactions are dedicated to capturing the energy of sunlight. That energy of sunlight is converted into a temporary chemical energy carrier, not directly into sugars, but into a temporary form. Those temporary forms are ATP and also another molecule called NADPH. If NADPH sounds familiar, that's because we saw a very, very similar molecule in cellular respiration called NADH, which was, as I said, an even more temporary chemical energy form than ATP. The only difference between NADPH and NADH is that the NADPH has one additional phosphate group, which is where the P comes from. But its purpose is the same. It carries a very temporary form of energy. We can also see that in the light reactions, water is taken in. Of course, plants get that from their environment, and they convert that into oxygen, which is released in this case as a waste product of the process. Let's talk for a moment how that sunlight energy is absorbed in the first place. The membranes that surround thylakoid discs contain many different pigment molecules. And a pigment is a molecular structure that is capable of absorbing some sort of light energy. There are lots of different types of pigment molecules. Here we see in this graph 3 of the most common pigments that are found in plants. And on the graph, it shows you a spike for the type of light color that pigment likes to absorb best. For example, we can see that chlorophyll A, one of the pigments, is very, very good at absorbing red wavelengths of light. We can see over here that carotenoids are very good at absorbing blue wavelengths of light. So my question for you in this next checkpoint, looking at this graph, what colors of light energy are not absorbed by these common plant pigments? And then speculate, what is it that happens to this light instead if it is not being absorbed and used for photosynthesis? So to summarize what happened in the light reactions, which is the first of the 2 steps involved in photosynthesis, we saw sunlight energy captured and converted into chemical energy in the form of these 2 temporary energy carriers, the ATP, which you can see right here, and the NADPH, which you can see below it. ATP and NADPH are going to carry that sunlight energy into the Calvin cycle. And the Calvin cycle is going to use that energy to assemble molecules of carbon dioxide into molecules of sugar. This is happening in the stroma of the chloroplast, so it's happening in the dead space around the outside of the thylakoids. And the magic of it is that the plants absorb this CO2 from the atmosphere, and they actually use that as the building blocks to make sugar molecules. So next time that you're eating sugar, think about the fact that it used to be a gas. It used to be CO2 that some plant absorbed. In the process of doing this, they convert the ATP into its uncharged form of ADP, and they do the same with the NADPH. It turns back into NADP+. These are the uncharged forms of the molecules, but remember, the beauty of them is that they can be recharged, and so they can travel back to the light reactions, pick up more sunlight energy, and in a cyclical manner, drop it back off at the Calvin cycle before, again, being decharged, and the process repeats over and over again. So to polish off this lecture, we have a few more checkpoints here to complete. Here you can see the overall recipe for photosynthesis, and what I want you to tell me in this checkpoint is in which of the two stages is the oxygen released? Which of the two stages generates that oxygen as a waste product? And then finally, last checkpoint, I have two organisms here on the slide, and I want you to classify each as either an autotroph or a heterotroph, and tell me whether they perform cellular respiration, photosynthesis, or both. And when you're finished with this checkpoint, you are finished with Chapter 4 and Chapter 5 lecture.